Many of you joining us will already be fellows of the Society. For those who are not members yet, please do consider joining us. The vision of the Linnean Society is a world where nature is understood, valued, and protected. We encourage debate and discussion about natural history, bringing together diverse communities and engaging with scientists, historians, artists, and all those interested in nature. This evening, I am delighted to welcome Professor Beth Okamura, who's going to be talking to us about her lifelong fascination with unusual and non-charismatic animals, fostered first by encountering colonial invertebrates called bryozoans on the Pacific seashore, and later by the serendipitous discovery of Myxozoan parasites in freshwater bryozoans in Oxford. Her varied research focuses on interactions between bryozoan hosts and Myxozoan parasites, dispersal of freshwater organisms, drivers of diversification, transitions between freshwater and marine environments, and extinctions of affiliate taxa, parasites in threatened hosts, amphibians. She is lead author of the highly regarded first textbook on Myxozoans, Myxozoan Ecology, Evolution and Development, and she's a merit researcher at the Natural History Museum in London. So without further ado, I'm very happy to pass over to Beth and looking forward to listening to your talk this evening.
Thank you. Okay, I need to share my screen. Well, thank you very much for joining me, all of you out there, to hear my talk, which is entitled The Hidden World of Parasites. Here's an overview of what I'm going to tell you about today. First of all, because a lot of people probably won't know very much about parasites, I'll do just a very brief general introduction. And then I'll go on to focus on just amazing radiation of endoparasites called Myxozoans, which is the group that I've been working on for a number of years. So I'll talk to you about what they are, because that's been mysterious for a long part of the time they've been known, what it is that they do, and the sort of staggering diversity that we're now discovering about these parasites. And then I'm going to close by considering the importance of serendipity in doing research, and how stumbling upon things is such an important way of getting to really grips with the underlying causes of things and leading to scientific discoveries. So what are parasites? Well, just generally, I'll let you know that parasites are a very diverse and ubiquitous group of animals. There are something like 43% of the animal phyla contain parasites. And you can see this is eggs of flukes. Here are lampreys and fish. These are protozoan parasites. There's a tapeworm, a tick that many of you will probably be familiar with, and things like ringworms, nematodes. So there's a vast diversity of parasites, and they've evolved independently a couple of hundred times within the animal group. So they're a diverse and varied group of organisms. But these are really very minimal estimates, because parasites are extremely poorly sampled relative to free-living animals. They're also very ubiquitous. And so that ubiquity means that they're important in things like energy turnover, food webs, and then leading on to ecosystem function. So for example, this is the life cycle of a parasite that infects fish, and these infected fish will be eaten by water birds. And, so they're important as prey. Parasitized animals are important as prey animals to other animals. And these kinds of interactions can lead to parasites being very abundant in food webs. So here's a food web, and each of these little balls represents a species in the Carpinteria salt marsh in Southern California. And you can see that the yellow ball and the lines link who eats who in this food web. And you can see that there are quite a number of yellow balls in this food web in a salt marsh. So they're diverse and ubiquitous and important for ecosystem function. And they also have very diverse life histories. So those life histories range from simple to complex life cycles. And that means that simple life cycle parasites, for example, would go from human to human or from another animal to another animal, but individuals of the same species. Whereas parasites that have complex life cycles will involve parasites that use, for example, a bird host, and then they go into snails, and then back to the bird host. This is an avian schistosomiasis life cycle, and occasionally those little transmission stages can be mistakenly transmitted to humans, and that's why you have humans in this life cycle here as an accidental offshoot. So you have these simple life cycles directly between one individual to another individual of the same species, or these more complex life cycles. And then you can also have parasites that are generalists and specialists, so that at any one point in these life cycles, they may be able to stage in the life cycle, they may be able to infect multiple different species or just a single species, in which case they'd be specialists. And then another aspect of parasites is that they can be ectoparasites, for example, like ticks, or like these isopod parasites on the external surface of a fish and living within on the tongue, or they can be endoparasites, like these nematodes that live in the digestive tract of a fish, or these eggs of a fluke that have been laid down in the muscle tissue of the host. And when parasites are endoparasites, they are especially hidden from view, of course, because they're living on the inside of the host. And that really even further hinders our understanding of parasite diversity and ubiquity and so on, because everything's hidden from view. So it's harder to understand how diverse they are, what their ecology is, and to understand their evolution. Okay, so that's a general background to parasites. And what I want to do now is to go on and tell you about myxozoan parasites, which are the group that we're going to focus on today, because I've been spending a lot of time over my research career studying the biology, ecology, and evolution of these incredible endoparasites. So they are endoparasites. They live within other hosts. They were first described from tissues of whitefish, a species which is now extinct, in Lake Geneva in 1825, where they were discovered as cysts. For much of the time the Myxozoa have been recognized, they've been regarded as protists, so single-celled animals, single-celled organisms, and that's due to the cellular nature of many of the stages that have been studied over a long period of time. And it was also recognized for a long time that they produced spores internally within these cellular-like structures called zoosperms originally. Myxozoa cause a number of important fish diseases, for example, whirling disease, proliferative kidney disease, which you'll hear more about, a soft flesh disease, which you get cysts and muscles and make them look untidy and not very appetizing. Myxozoans, however, also exploit a diversity of other vertebrate hosts. So in addition to fish, they're found in reptiles, turtles and snakes and so on, amphibians, here's a snake, and in also in birds and in small mammals like shrews and moles. So an amazing diversity of vertebrate hosts that we're now discovering that myxozoans are associated with. They have complex life cycles, and this is a life cycle of sort of generic life cycle to show you. So they infect these vertebrate hosts, but they also have infection stages in invertebrate hosts. And between these two different vertebrate and invertebrate hosts, you have waterborne spores, and these are the transmission stages. So spores are released in the water, and those enable the parasites to infect the next host in their particular life cycles. They were recognized, this complex life cycle was recognized only relatively recently, because as I say, they were discovered in the 1800s, but not recognized to have these complex life cycles until 1983. we now know that they infect a range of hosts in freshwater, marine, and terrestrial environments. But most of the species that we recognize today are described in fish. And as you can see, there are two main invertebrate groups so far recognized: the annelid worms and freshwater bryozoans, which are the group that I've mostly been working on. Okay, but what are myxozoan parasites? So I mentioned that the cellular nature of these plasmodia, which were really the first stages that were discovered and detected for a long period of time, that sort of cellular nature suggested that they were protists or protozoa. But the clue that they were potentially metazoans was recognized fairly early on, because the spores, these infective stages that you can see here, were recognized eventually to be multicellular. And then much more recently, with the discovery of sacs and myxoworms, and I'll tell you more about that because I've been involved with that, these are definitely multicellular stages. And so these signals for multicellularity then suggested that actually myxozoans are metazoans of some form. So these are proper multicellular animals. However, cnidarians, which are things like jellyfish and sea anemones, were actually suggested to be what myxozoans really belong to as far back as Weil in 1938, because he noted that the intracellular organelles that were recognized in spores, called polar capsules, were very similar to nematocysts of cnidarians. And you can see, I think, that both of these forms have these coiled up filaments that go shooting out. For cnidarians, they use them for prey capture, and for myxozoans, they're used to attach to the next host. So there was this suggestion that they were cnidarians quite early on. But for a long time, they were recognized as, they were just, wasn't clear what they were. So I don't know, can you guys see this thing at the top? Not really.
No, we can't see anything.
I've got this thing saying you're a screen. Anyway, it's okay. I guess you probably can't. So I'm now going to tell you about the molecular insights that have allowed us to really pin down the cnidarian affinities of myxozoans. And these are based on looking at DNA sequences in phylogenomic studies. So where we are able finally in the past recent decades to really get a handle on multiple genes in the genome that enabled us to pin down the affinities of non-model organisms, as it were. And so I was involved in that first publication, really, that showed you that Budimbrachia, which is one of those myxoworms that I showed you, that Budimbrachia really is an cnidarian worm, and I published that with colleagues in 2007. And then subsequent to that publication, there have been further publications really confirming that on the basis of phylogenomic analyses, that myxozoans are definitely cnidarians. We also have evidence that myxozoans have cnidarian specific genes, and these genes are diagnostic for proteins that are used in nematocysts, which are those intracellular organelles that are highly unique to cnidarians. And so we found, for example, that there's a novel mini-collagen gene. We were the first ones to show that myxozoans actually have mini-collagen genes, and these link them with cnidarians because they share these polar capsules, which are basically the same thing as nematocysts. So those sorts of evidence then really now have pinned down that myxozoans are a radiation of endoparasites within the phylum Cnidaria. And as I said, cnidarians include things like sea anemones and jellyfish. So before myxozoans were recognized as cnidarians, cnidarians were all free living forms. So they were the anthozoa, jellyfish, corals, and so on, their relatives, and the medusozoa, which are various jellyfish and hydroids and so on, all free living cnidarians. But with the recognition that Myxozoans are actually cnidarians, we have a completely kind of new view about cnidarian biology, because we have this big group of cnidarians that are endoparasites, the Myxozoa, and their sister taxon, Polypodium, which is so far only a single species that's been described, paddlefish and sturgeon. So the Myxozoa are a big radiation, and I'll tell you more about their diversity a bit later on. But the inclusion of these endoparasites in otherwise free-living cnidarians is, for invertebrate zoologists, just an amazing new development. And collectively then, these parasitic cnidarians, I forgot to mention Polypodium is a parasite. They form what we now call the endonidozoa, along with the anthozoa and then medusozoa within the cnidaria. So a little bit about what Myxozoans do, their biology. So I mentioned that they use these multicellular spores that have polar capsules. And here you can see four polar capsule in a multicellular spore. And these polar capsules have these filaments that go shooting out. And here you can see one that's shot out. And they allow Myxozoans to attach to the next host in the life cycle. Myxozoans develop as these multicellular sacs or myxoworms, as I mentioned, or in unicellular plasmodia like these. But the strange thing about these plasmodia is that within these single cells, you can have 1 to hundreds of cells developing within the cytoplasm, which is kind of an unusual thing. The development of Myxozoa involves the formation of cysts, for example, in tissues. These are cysts in the gills of dace. Here's cysts in a swim bladder of a perch. And other Myxozoans can develop in hollow spaces. And here's a nice example of that. You can see these sac-like forms developing in this hollow body cavity of a freshwater bryozoan. You can see all these tentacles of the bryozoan out there, eager to feed on plankton. Okay, so Myxozoan infections, what do they do? Well, generally, they're pretty innocuous. So they don't tend to make their hosts, when they're infected, look as if they have any disease whatsoever. So infected hosts generally look quite healthy. However, there are some Myxozoans that can cause severe and emerging diseases. And here's a table of 8 emerging diseases that are recognized now worldwide. And these emerging diseases are all involved with fish, and they are variously linked with things like climate change, if the parasites were introduced to new regions, if the environment is changing because of alterations like putting in dams, for example, or when you're trying to raise a lot of fish under stressful conditions when they're very dense in an aquaculture kind of setting. So that's where we recognize these emerging diseases. But recognizing emerging diseases can itself be challenging, because you have to regularly monitor the fish hosts. And if these diseases are happening in wild populations, detecting infected hosts can be difficult, because if they die, they're very often scavenged by wild animals around. So these are sort of challenges that we have for detecting emerging diseases in general. Some of these diseases are important for aquaculture and fisheries, and these I showed you earlier, where you have a whirling disease in salmonid fish, proliferative kidney disease also in salmonid fish, and soft flesh disease in wild fisheries that are important. So now I'm going to move on to proliferative kidney disease, which is an emerging disease that I've been working on, abbreviated as PKD. And this is a disease that's caused by a myxozoan that develops in freshwater bryozoans. And all salmonids seem to be susceptible to this disease. And its source was, for a very long period of time, a great mystery. It was known to be caused by a myxozoan, but where the disease came from wasn't known. It causes a lot of economic loss in aquaculture. So for example, in 2018, 70% of trout were lost from pens in Pacific seafood in the Columbia River and in Washington. And it also was responsible for really huge fish kills in the Yellowstone River in Montana, which is a big river where you've got, fly fishermen going out to fish for salmon and trout in the river, mostly trout actually. And this caused a large economic loss. They closed the river for the whole of the summer and so on. And so I come to the picture because essentially I and colleagues along with their help was able to show that the source of PKD is freshwater bryozoans. So I found that freshwater bryozoans contain sacs of Myxozoa. These sacs develop spores, and those spores are released, and they are released to infect salmonid fish. And then the fish in turn release spores that infect bryozoans. So we first showed that we get the same parasite based on molecular data in the fish with PKD and the bryozoan hosts. And then we were able to show that, yes, you can transmit the parasite back from infected fish to bryozoan and show that this closes the life cycle of this parasite. So we were able to then show this is, where this big disease in aquaculture is actually coming from. Following that discovery then, I then went on to look at some of the strategies that the parasite uses to exploit the invertebrate hosts, these bryozoan hosts. And what we've been able to show on the basis of a lot of field work and lab experiments and so on, is the parasite cycles between what we call avirulent and more virulent infections in the bryozoan hosts, and that's driven by host conditions. So when the bryozoan is in very good condition, you get these massive numbers of sacs developing. You can see these branches of this bryozoan colony filled up with these sac-like things. And in turn, those sacs have hundreds and hundreds of spores that are released to infect fish. And that happens when the bryozoan is really vigorous and in good condition and can support that massive development of a parasite. But when the bryozoan becomes less healthy and in poorer condition, that triggers the parasite to then revert to single cells as covert infections that are pretty much avirulent. They don't impose any impact on the bryozoan host that we can see. And so when you have that kind of cycling going on in our bryozoan populations, that will then promote a long-term persistence of the parasite in these bryozoan hosts because it's developing dependent on host condition. And that means that you have this long-term disease reservoir for fish that are going to pick up PKD in aquaculture or in the wild. We've also been able to show that the parasite is able to infect stadoblasts, which are the dormant dispersive stages of freshwater bryozoans. So this is a cartoon of a freshwater bryozoan. They produce these seed-like stadoblasts, which are little tiny, about 1 millimeter seed-like stages. And those in turn a number of studies that we've been involved with show that those, for example, can be ingested by birds, and those birds can transport infected stadoblasts to other sites. So it's a whole kind of mechanism for the parasites then to be dispersed to other places and potentially introduce the disease to new environments. Okay, back to a bit more about the biology of myxozoans then. Some notable features that they present to us are linked with being endoparasites. So they show a very high degree of morphological simplification relative to their free-living cnidarian relatives. And hopefully you've been kind of picking up on that as I've gone along. So, they don't have tentacles, they don't have a gut, they don't have a nervous system, they don't have any recognizable gametes. And these are all things that their free-living relatives do have. Also, if you look at the cellular level, they have no cilia on the cells, and they have no centrioles, which are intercellular structures involved with cell division, for example. And these are all features that their free-living relatives have within their cells. So another big notable feature too is that these endoparasites are very miniaturized. So free living relatives of myxozoans, jellyfish and sea anemones are tens to many tens of maybe even hundreds of centimeters across in some cases. Whereas parasitic myxozoans, myxoworms are 2 millimeters, sacs are 0.2 millimeters, and plasmodia are again very small. So miniaturization is another notable feature that myxozoans demonstrate. And these very simple morphologies and small sizes are things that are often linked with parasites, but myxozoans really take these kinds of trends to the extreme. Okay, so there are two major groups of myxozoans, the Malacosporia and the Myxosporia. And the Malacosporia is a group that includes those multicellular sacs and worms. And the Myxosporia is what we call those myxozoans that form these unicellular plasmodia. And within both of these different types of myxozoans, we have spores that develop internally within a multicellular form or internally within a unicellular form. And these are spores here. So I've been mostly studying the Malacosporia, myxoworms and sacs. And these are some of my colleagues, Liz, Alex, and Hannah, who've been really pivotally involved with my work in this area. So I don't have time to tell you about, all of the things that we've done, but we've looked a lot at the development, the diversification of Malacosporia, early development, systematics, describing new species, new orders and classes. And this is that textbook that was mentioned in the introduction that I've been involved with putting together. But what I do want to tell you about and spend a little time is the unique motility that Malacosporians show. So sacs are inert and myxoworms are motile. And their motility is driven by muscle tissues. So this cartoon here is shown in ultraspectra here, and these four little segments here are muscle blocks, 4 muscle blocks that are present in myxoworms. And the green staining here in this confocal micrograph shows you those longitudinal muscle blocks running along the whole length of the myxoworm, this Buddenbrockia. And I'm now going to show you the amazing locomotion that those muscle blocks enable in myxozoans. So this is Buddenbrockia swimming. That's a 1 millimeter grid in the background. And this is just slowed down in a highly viscous solution. So these guys are very capable of locomotion in a completely unique way. No other cnidarian, no other worms locomote like that. And that basis of the motility is based on the body plan of the free-living cnidarians, which involve a gastrodermis and an epidermis, 2 epithelial layers, inner and outer epithelial layers that are formed by epithelial muscular cells. So you have these cell bodies with muscle fibers that extend out along these 2 layers. And what Buddenbrockia does is those 2 layers, the muscle fibers themselves actually disattach from the epithelial layers and form these long longitudinal muscle blocks. Okay, I want to now move on to looking at some other amazing worm-like motility, but this time shown by plasmodia, these single-celled Myxosporia. And this is in conjunction with my Brazilian colleague, Edson Adriano, at the Federal State University of Sao Paulo. And so these are myxozoans that are unicellular. This is what they look like. They have actin filaments. We've shown them by ultrastructure, these sort of net-like things here. These are present within an outer layer around an large internal vacuole within these cells. And here's a confocal micrograph with the green stain showing you the actin filaments that show up in these worms, in these worm-like single-celled things. So I now want to go on to show you some videos of how these guys move. Oh, and yeah, so we've published, how to build single-celled cnidarians with worm-like motility. So this is, these are single-celled ceratomyxid worms from bile bladder, from gallbladder fluid collected from fish from the Amazon. And this is the way they move. Some of them are filled up with spores. So you can see, you know, these sort of more densely filled ones, you can see the spores inside, and the ones that, you know, look more clear are not yet mature and don't have the spores. So, you know, these guys are moving around in the bile duct, and they are capable of translocation. Oops, I need to go on to the next slide. Here's another vermiform plasmodia, a species that has more blunt tips, and you can see some spores developing inside. So we've, you know, there are now a number of species that we've discovered that take various slightly different morphologies and undergo that amazing locomotion, showing a worm-like locomotion, but at a single cell level. So myxozoans demonstrate two paths to build a worm. And so they can converge to worm-like forms like earthworms and nematodes and other multicellular worms, either via longitudinal muscles as myxoworms, or by using actin filaments in a single cell by these vermiform plasmodia. So these two absolutely amazing ways to converge to worms, but in completely unique forms. So, as I say, totally unique forms, but the million-dollar question now is, why be motile? Well, that we don't know yet. We have some ideas, but it's a big question. Okay, on to myxozoan diversity then. And this is work with Ashley and Juliana. And so let's go and consider how many myxozoans we have evidence for. Well, at present, amazingly, about 20% of extant cnidarian species living in the present day are parasites. I mean, that is staggering. We didn't even recognize that myxozoans were cnidarians until, you know, the last decade or so. So 20% on our most recent count are of cnidarians are myxozoans. But this is greatly underestimated. Myxozoan species accumulation curves are not saturating. So, you know, the number of species being estimated over time is going up. Here's our recent estimate. So it doesn't look as if there's any plateau for global species richness. And nor, if you look at Brazil, is there any evidence that the number of species described over time is decelerating and plateauing off. So, you know, there's lots more species out there to be described. So yes, we don't know how many there are. There's also other evidence that the diversity is greatly underestimated in the form of novel sequences that are being encountered by us when we metabarcode environmental samples of DNA. So you can take water or tissues and filter that water or extract DNA from those tissues. And this is, most of this has been done in very well sampled regions. And what we find is that only 7% of the sequences that we are finding in these studies can we link with sequences that are already, this is DNA sequences, already available in the data bank for those DNA sequences called GenBank. So 93% of our sequences are novel sequences, which is pretty amazing. So those kind of data then really suggest the possibility that, you know, myxozoan diversity is really so far underestimated. And it might even be that we have more parasitic cnidarians than free-living cnidarians. And so, for example, Naldoni et al. Speculated in a paper in 2011 that there might be potentially 6,000 species of myxozoans in neotropical freshwater fish, if on the basis of their findings, you might find about 2 myxozoans in each fish species. So 16,000 species is way more than, you know, we presently have in the free-living cnidarians. Okay, so on to lessons in serendipity then. So I hope I've kind of made the point to you that hidden parasites can be difficult to study because they're not evident. Also, I pointed out earlier that myxozoan infections are typically innocuous. So even detecting that a host is infected can be difficult. So they don't look as if they're infected. Infections also are typically patchy. So you might find, it might be the case that, infected populations are found here and here, but not anywhere else in this freshwater habitat. So you have to be lucky to find the right place to find the parasites. And these sorts of issues mean that it can be a challenge to resolving complex life cycles, because you have to find the infected host potentially from the vertebrate and the vertebrate stages. So these sorts of issues make, finding parasites and finding infected hosts difficult. So insights can rely on luck, and you might find that they lead to unanticipated avenues and other inputs. So I now want to kind of take you on my own serendipitous journey of working on myxozoans. So I really discovered myxozoans during an incidental discovery of myxozoans and myxoworms and sacs. So I was studying freshwater bryozoans, and this was driven by curiosity-driven research, because I was interested at the time in looking at research and looking at how freshwater organisms are dispersed by waterfowl. And I used freshwater bryozoans as a kind of model system to look at that. So I was collecting bryozoans and looking at them with a microscope because I was interested in production of statoblasts and reproduction and so on. And then I just started seeing these weird worms inside. Here's a myxoworm sticking out next to the tentacles of a bryozoan, and these weird sacs and bryozoans, and going, God, what are these things? And, looking around for parasitologists, because I was not a parasitologist, to help me figure out what they were. So that was, how we discovered this whole group of myxozoan parasites in freshwater bryozoans. So that initial discovery of sacs and worms then led to confirming the cnidarian status of myxozoans. It involved describing the major new clade infecting freshwater bryozoans, the Malacosporia, which is a very primitive clade compared to this much more derived Myxosporia, the thing that produces these plasmodia that had been recognized for so much longer. That discovery of sacs and worms also led to detecting further novel myxozoan diversity. So for example, the diversity we find by doing metabarcoding of environmental samples and all these vermiform plasmodia that we're discovering. And it also led to the discovery of the long sought source of PKD by finding infected populations of freshwater bryozoans that then go on to infect the fish and in turn reinfect those bryozoan populations. So that then led to the long-term studies on the ecology of the causative agent of PKD that I told you about, and the impacts on the bryozoan hosts, which in turn then led to insights on epidemiology in fish diseases. So for example, our understanding that we have these host condition dependent development of these sacs within the bryozoans that then go on to release spores when the bryozoans are in good condition, when they're really growing vigorously in the summer, when temperatures are warm, when there's a lot of food around. That then explains when it is that fish come down with PKD and develop these severe cases. So understanding the ecology of the parasite in the invertebrate host then lets us to, have much more insights into the epidemiology of the disease in their fish hosts. So hidden parasites and take-home messages. I hope that I've kind of been able to show you an example of how my work has led to insights into how the world is not always as it seems. We didn't know that cnidarians had this huge diversity of endoparasites until I happened to stumble across, this weird group of parasites that infects freshwater bryozoans. These unanticipated findings can lead to lots of different breakthroughs as they, cascade down. And in turn, I'm hoping that I've shown you how curiosity-driven basic research that initially was driven by my great interest in how freshwater bryozoans disperse from site to site, because they have to be, many of animals that live in freshwater habitats are unable to get up and fly or move to another place. They've got to be taken there by something like birds or fish or whatever. So that was the initial stimulus to study how birds might be carrying around bryozoans that really led to this kind of cascading series of discoveries that I've been telling you about today. So I'd like to thank you for listening, and to thank the Linnean Society for inviting me to give this talk. I've been very lucky in having lots of research funders over the years who've enabled me to gain these various insights into the systems that I've been telling you about, and many, many colleagues, some of you whom I've shown you as I've gone through this talk. And so that's it. Thanks for your attention. And I think maybe we're going to have some questions now.
Hello. Thank you so much, Beth. That was really interesting, fascinating talk. And we have some questions for you in the Q&A. And if anyone else in the audience has any questions for Beth, please pop them into the Q&A now. So first of all, we have a question from Joe Long, who asks, is there any evidence of them occurring as an emerging disease as the result of the arrival of invasive non-native fish species? Thinking of the situation in Norway, where Pacific salmon escaped from fish farms in the north of the European continent and have established in Norwegian rivers. Could these fish have brought the parasites, which have then jumped to native salmonoids?
It's a possibility. The very interesting thing about the emerging disease, and PKD, of course, is the system that I really know the most about, is PKD is an emerging disease in aquaculture systems here in the UK and Europe. And that is because those systems, I think, and many of my fellow colleagues would agree with me, that seems to relate to the fact that aquaculture involves introducing rainbow trout, which evolved in North America, to a European setting. And those rainbow trout are non-native, and they're getting infected by European and UK strains of the parasite that causes PKD. Okay, so they're picking up a parasite that they haven't evolved with, and that might be part of the reason that the disease is so severe in introduced rainbow trout in aquaculture settings, because the native parasite in a non-native fish host haven't co-evolved over a long period of time. So you get a very massive kind of, immunological response of the fish host to the presence of the parasite. And it's that response that causes that kidney swelling and PKD development. The disease in North America, whirling disease in North America, seems to have been arisen when European infected fish, if I think I have the story right, infected fish from Europe were used to feed salmon in North America, or trout, I can't remember which one, in aquaculture. And those infected fish then somehow managed to introduce the myxozoan that causes whirling disease to North America. And that might have contributed to whirling disease as being an emerging disease in North America. So I don't know of any examples, but those were frozen fish that were fed. I don't know of any examples. There might be some out there of wild fish or non-native fish being introduced somewhere else and then importing a myxozoan with them. So that's kind of a long answer to the question as I understand it.
No, that sounds great. So yeah, it's a distinct, possibility in the future as well that it could happen.
Yes, exactly. Yeah.
Yeah. And still on the subject of emergent diseases, Ellen Hildrew has asked, how convinced can we be that these are truly emergent diseases rather than diseases that have always been out there, but we're just coming up against them because of conditions in aquaculture?
Yes, that's a good question. I mean, so I think there's good evidence, for example, in Switzerland, that the Swiss are they're very proud of their supposedly wild rivers and so on, and they have many fisheries biologists who've been sampling there for many decades annually their brown trout populations, and over many decades of data, they now have accumulated enough data to really pretty clearly tie down the fact that PKD is an emerging disease of the wild brown trout populations, and that this is linked with warming waters and eutrophication and so on. So the PKD is now emerging in a number of other European countries in wild fish populations in Germany and Austria, and in some of the, in these sorts of places where there's sufficient data now to suggest that, we are getting an emerging disease situation, and that the fish kill that I mentioned in the Yellowstone River, that also would have been driven by native parasites in native cutthroat trout populations that just had a massive fish kill, and it was so massive that everything wasn't scavenged, and the fisheries biologists were picking it up. So I'm kind of exploring around my memory of the different scenarios to try to answer that question, but hopefully I've gone a little bit towards that.
Well, thank you. I'm going to move quickly over to the chat because there's a related question here. So if animals or other humans eat the fish that are infected, does it cause them any problems?
Sorry, can you repeat that?
If animals or if humans or other animals eat the fish that are infected, does that cause any problems?
There's no evidence that PKD, for example, is transmitted to humans if they eat fish that are infected. There's only one case where there may be fish-to-fish transmission, and that's for one species of myxozoan called Endromyxa. But otherwise, the life cycle is a complex life cycle that requires going from the fish to the invertebrate host and then back to the fish. And the case where we know that there seems to be fish-to-fish transmission was worked out in an aquaculture setting in the Red Sea, I believe, by an Israeli group.
Cool, thank you. And then we have a question by David Rollinson here, who says, interesting talk, thank you. What is the life cycle of myxozoans involving moles with no stages in water?
Oh, it's an excellent question. You know, the life cycles, while we have, many thousands of myxozoan sequences, or many thousands of myxozoans described, and many more sequences of myxozoans out there in the database, there's only something like 35 life cycles that have been resolved. So we have a tiny handful of life cycles resolved. The life cycle for the myxozoans that involve moles and shrews is super interesting, and I'm sure that it must involve oligochaetes, earthworms, because these animals eat earthworms, and earthworms are annelids. And I think a few people have fiddled around, and including people in my group, of seeing if we can get any positive infections in earthworms. But as I say, infections are very patchy, and you get negative results, and you don't know how to interpret that. But it could be opening up a can of worms, as it were, because if earthworms really do carry infections, shrews and moles giving us a signal that they may, then other things that eat earthworms, like blackbirds and hedgehogs and lots of things, might be hosts for myxozoans. But infections are innocuous. They just aren't probably being sampled even through pathological investigation, because nobody thinks to look for them. So it's a really amazingly open, interesting question that hopefully someday somebody will resolve.
That's amazing. It feels like, you open a little door onto one world, and then suddenly there's all these other doors and avenues which can be explored from there, which is really fascinating. A question from Steve Trim now. Amazing talk. Do you know how these plasmodia go through mitosis without centrioles?
I can't answer that question, but all I can say is that they do, because the cells divide, and we also know they undergo meiosis, because we can see meiotic divisions as well in ultrastructure. Yeah, I don't know. It's a good question.
It's another avenue for study, definitely.
Yeah, I think there are a few other groups that centrioles are lacking in. So it may not be completely unique.
Interesting. So the kind of the version that we learn maybe in A-level biology with the centrioles, maybe it's all a bit more complicated than that.
Yeah.
I'm going to return to another question from Alan Hildrew. So does PKD infect Atlantic salmon or only Pacific salmon, including rainbow trout? Brown trout is now extremely widespread, e.g. South America, Africa, New Zealand. Does it transmit parasitic myxozoans?
PKD, according to the fisheries, parasitologists and biologists and so on, all salmonids are susceptible to PKD, first of all. So yes, Atlantic salmon should be able to carry an infection. I have wondered about whether brown trout, the introduction of brown trout to places like Chile, could introduce the parasite to Chile. That would mean the brown trout would have to excrete viable spores that would then have to infect local bryozoans. And those local bryozoans would then have to be competent hosts for the disease to become endemic, as it were, in places where brown trout have been introduced. I think that, when brown trout are introduced to places, I would think that the people who are introducing them have some concern that they're not introducing parasites along with the fish that they're introducing to these places. So, maybe they are screening them to some extent. But I also see that it is a real possibility that could be a scenario. So far, I haven't heard of PKD in places like New Zealand or Chile, you know, where you do have brown trout being introduced for fishing.
So something where the sort of, you know, the vigilance of looking out for these things, along with many other sort of emerging diseases, is really critical.
Yeah.
And I'm going to ask a question now, which is our last question at the moment up on the board, because you mentioned, you showed us these two really interesting ways that they were showing motility with such a simple organism. And you said, why be motile? And said, this is really interesting. We don't know why, but you have some ideas.
Yeah.
So would you be able to explore some of these ideas?
Yes.
Of what
It's just crazy, you know, that you saw they are really very motile. And so one idea that I sort of favor in both the multicellular myxoworms and the plasmodia is that if you're living within a fluid-filled cavity of your host, which is what they both are doing. So the motile myxoworms are in the fluid-filled cavities of bryozoan hosts, and you can see the infected bryozoans, and you look through and you can see them going like this. And you saw that the little unicellular plasmodia in the bile were doing the same thing. And what that movement might be doing is breaking up the boundary layers that would be developing around the organisms, because basically these endoparasites are going to be taking up nutrients from these fluid-filled spaces that they're living in. So they're getting their food by taking up nutrients from these fluid-filled spaces. And they're also, of course, going to be having some excretory products as well that they'll be excreting. So if you're moving like this through that fluid, you could be breaking up the boundary layers a little bit and helping these, you know, reduced layers of thick layers of more stagnant water that develop around surfaces. So if you move and you break up those layers and thin them down, then that might be enable nutrient uptake to be more, you know, more efficient than if you're just sitting there trying to, you know, immobile. So that could be one reason. We speculated for the worms living in the bile duct, you know, you are going to have bile being excreted. So maybe it keeps them in situ within the gallbladder, and so that they're not excreted before they're ready to be excreted, or presumably they eventually are, because they've got to get rid of those spores and into the environment to infect the worms or whatever it is in the next life cycle. We think they're probably worms. So that might be another explanation. The amazing thing about the myxoworms is that, you know, the movement that you were seeing was of a worm that had been released from the bryozoan host. So you can squish a bryozoan and the worms come popping out. And then they're just amazing. They just move all over the place. And eventually they get tired and they sit in one place and kind of weakly corkscrew, but then they, you know, and they move around again. So I kind of wonder whether, and we also have seen, I mean, I showed you that image of the myxoworm kind of sticking out of the bryozoan. The tentacles of the bryozoan were sort of, you know, here, and then the myxoworm was sticking out further. So they can escape from the bryozoan host. So maybe if the bryozoan is being eaten or something like that, then the myxoworms can escape and swim up into the water column. And, you know, they're being a couple of millimeters long. They might be eaten by a fish, and therefore they might, if they've got spores that are infected to fish, then that might be another way that they can infect fish rather than wait to be released as spores from the bryozoan host. So, you know, there could be multiple reasons for motility. But, you know, the weird thing is that the sac-like relatives to the myxoworms are about the same diameter as myxoworms. And so if you're just simply relying on diffusion to get all your nutrients in as a sac, and you're not moving around, then why can't you do that if you're a worm? Because they're of a similar diameter. So, you know, it's really, it's a big question, and we don't know. We can arm wave.
And is it something that you will investigate some more in the future of some of these alternative scenarios?
Well, maybe not me. Maybe somebody else will happen to stumble across. One of the things that's quite difficult is actually getting material to study, because you need to be in the right place at the right time, and prevalences can be quite low. But and then you've got, you know, we don't have a culture system that works well with you know, raising these things. So it depends on collecting from the wild and doing quick studies, you know, with wild collected material. So we don't have the luxury of just being able to breed them in the lab and study them. So there are lots of difficulties to getting at those questions, but, you know, hopefully someday somebody will have some serendipitous insight.
Oh, well, fantastic. Thank you so much. Thank you for a fascinating talk about your own research, for highlighting to all of us the importance of serendipity in our research, and some interesting avenues for other people to follow as well in terms of research ideas in the future. So just a big thank you from me and everyone at the Linnean Society, and everyone in the audience for joining us this evening. Thanks, Beth.
Thanks very much.
Bye.
Bye-bye.